Most of this you will already know. The first point perhaps is worth emphasizing. Most trematodes, including most blood flukes, are hermaphroditic. So a single individual contains both male and female systems. The only exception is one group of blood flukes, which are the schistosomes. Adult worms live in the circulatory systems of vertebrates. Eggs hatch in water, either in the sea or in fresh water, and larvae penetrate usually a snail, leading to the production of cercaria. You know that. They're normally very specific for the intermediate host. So the general rule is that one species of schistosome or whatever can only develop in one species or a few related species of snails. This turns out to be true at a fine scale, but at an evolutionary scale, it's not true. Cercaria penetrate through the skin of the final host. There's no second intermediate host in the blood flukes. Cercaria come out, swim in the water, penetrate you. And the reason for the very pathogenic effects that they have on the host is often due to the accumulation of their eggs in the tissues of the host. The presence of the worms by themselves appears to be less dangerous than the buildup of eggs through time. And you have all seen, I'm sure, many images like this. So there's a schistosome cercaria attached, there's the head of the cercaria attached to the skin, I think, of a mouse in that case. The head will break off from the tail and penetrate as a schistosomulum into the tissues. Well, the classification of blood flukes used to be very simple until last year. There were three families: the Apocalids in fish, the Spirochids in reptiles, and the schistosomes in birds and mammals. And only the schistosomes have separate sexes. That was until last year. The trouble is, some people in the US have started exploring this family, the Spirochidae, in more detail, and found a huge diversity of these things, mostly occurring in sea turtles and freshwater turtles all over the world. Nobody has really looked in Thailand, but they must be here as well, probably quite diverse as well. Now, these people have split this one family into 6 listed here. And you can probably tell the politics of the people who named this particular one, Barak Trematidae. But I'm not going to go into great detail on that topic. I'll go through the Apocalids and the Spirochids a little bit and just mention a few points about them. The Apocalids occur in fish, yes. They're usually fairly tiny worms, maybe up to 5 millimeters, but quite often smaller. And they live in some odd parts of the circulatory system. And they release eggs into the tissues. Eggs will build up in the gills of fish and in other tissues. In South Australia, there are cages set up in the sea, huge cages in which tuna are grown. This is called ocean ranching. And this is a very effective way of growing very large tuna. But the trouble is that you have a lot of tuna in one place, and the waters have to be fairly sheltered. It's not the open ocean. So it means that there can be a buildup of parasites in that area. And these Apocalids kill a lot of tuna and cause a lot of disease. And this kind of image is quite typical. This is heart muscle, and each of these granulomas you can see here surrounds a single Apocalid egg. These eggs also often build up in the gills. That's how they escape from the fish. So they build up in the gills and rupture the gills, causing damage there as well. Until quite recently, nobody knew what the intermediate host was, some kind of snail. Well, no. Does anyone know what this is? It's a terebellid polychaete, so it's a kind of annelid, a worm. It's not a snail, it's not a mollusk. These can get really quite big. The body can be that long. And the tentacles, these tentacles are used for feeding, and they can be that long. So sometimes you see these in the bottom of the sea. All you will see is an opening and then these striped tentacles extending. And the region they can cover can be like this. So they are quite large. And this is where you find cercariae of Apocalids. Interesting. And everybody knows that Apocalids only infect fish. Well, here's a problem. I found a species of Apocalid in the heart of a dugong. And a dugong is a marine mammal. What? Now that happened around the year 2000 because I was routinely examining for parasites and things all dugongs that were washed up in North Queensland in Australia. And I had my set protocol and I looked in various tissues and did various things. And I always checked the heart and treated the heart as though it was a turtle heart because turtle hearts are full of blood flukes. I thought, well, no one knows anything about what's in dugongs. I'll check dugong hearts. And as I was doing that, I saw half a worm. I thought it was half a worm. I thought that looks like an Apocalid, but Apocalids don't occur in mammals. Everybody knows that. So I picked it up with a pipette and then I managed to lose it. So I thought, it was nothing, couldn't be. And then I found a complete worm. It was an Apocalid. That's the front end of that Apocalid from a dugong. And we've also found quite commonly in histological sections in various organs of dugongs, we have found what look like schistosome eggs, but I think it must be eggs of these Apocalids. This turns out to be quite a common parasite in dugongs. In Thailand, you have dugongs. Parasites of those, I have no idea, but maybe it's here too. So this is a long-range host switch from a fish to a mammal by a blood fluke. Now you have a new species living in a mammal. I'll talk a little bit about the spirochetes, the turtle killers. So these are now split into 6 families. They are incredibly diverse in appearance, shape, life cycle, and in their DNA. And they're clearly very ancient. They have clearly co-evolved with the big turtles and the reptiles. Very typically, since I was also doing similar work on sea turtles, examining those for parasites when they were found washed up on the beach, every turtle I ever examined had spirochetes in it, these blood flukes. Quite often, when you open up a turtle, you see things like this. This is a packet, a little packet, a little almost like a granuloma, but I think not quite, of spirochete eggs on the spinal cord of a turtle. The damage that these things do to turtles is extraordinary. And every turtle anyone in Australia has ever examined has been infected. So as far as we know, they're only found in turtles. Virtually all are infected. And at least in parts of Australia, these spirochete infections are the main cause of mortality of sea turtles. Really, the main cause now is people. It's boats running over them. It's people digging up the eggs. It's people catching the turtles in the sea and eating them or whatever. And it's the eggs in the tissues that cause most of the pathology. Look at this. This is the brain of a turtle. There's a whole genus of these worms that lives in the brain. Very thin, fine worms. Look at the eggs. Look at the damage that's been done in that brain. And yet you can find turtles swimming around that appear to be quite healthy with the brains that look like this. These are the eggs of another kind of spirochete, very large. You can see them clearly with the naked eye. spirochete infection might or might not be associated with fibropapilloma, which is a herpes group virus caused by in sea turtles. And it's possible that spirochetes are actually acting as vectors in some way. This remains a little bit unclear, but it has been suggested that that's the case. So actually, this is not the same version of the talk. Oh, we'll see how we go. So indigenous people in Northern Australia eat sea turtles. So can people become infected with these things? So this is a local policeman in the Torres Strait Islands. His name was Piggy Nona. I said, Piggy? I can't call him Piggy. What's his real name? And they said, well, everybody calls him Piggy. And there was another policeman there, local policeman, whose name was TBS. I said, well, what's TBS? Is that short for Tiberius? No, it means too bloody stupid. I thought, well, I can't call him too bloody stupid, especially as he was that high and that wide. But they said, no, everybody calls him that, so you must call him that. So this is Piggy. And the people there chop up the turtles and they eat all the parts of it, including the gut. So this is the intestine of a sea turtle, part of the intestine. And those black speckles that you can see there are clumps of eggs of spirochetes. Sometimes when you open up one of these turtles and put your hand in there, it feels like sand. It's like sandpaper, it's gritty. And the grittiness is simply caused by the masses, the millions of spirochete eggs that are present. It's quite extraordinary when you see that. Also, the great vessels. I put another photograph in the talk, which seems to have not made it onto this version. Don't worry. But I found a photograph I've been looking for of the aorta of a turtle, the main vessel out of the heart supplying the body. And it's completely blocked by spirochete flukes. And yet the turtle could still function to some extent. So it's extraordinary the damage that these things can do. Well, around the year 2000, there was a scare in Northern Australia because eggs that looked like this were found in the feces of Aboriginal children. So the health department went into a panic and said, oh, we have an unknown parasite in Australia. What is this? So I had a look at it and said, well, it's very simple. These are spirochete eggs. Well, why are the children getting this? Are they infected? Well, no. I mentioned that the Aboriginal people in the north of Australia eat all the parts of the sea turtle, including the intestine. They just boil it and mix it with herbs and eat it. And these eggs are very tough. So the eggs are dead. They're in the intestinal tissues. The children or the adults eat them. The eggs survive passage through the gut. They're damaged, but they still are recognizable. That's why this is not a real case of human disease. That was the original panic paper that was written about these eggs in Aboriginal children. And I and a couple of other people then wrote another one to say, don't worry, be calm. They're not infecting the children. Then about 1990, freshwater crocodiles in the north of Australia were dying in crocodile farms. And the pathologists said there was some kind of parasite, but they couldn't identify it. So I spent a lot of time investigating that. That's a freshwater crocodile. They can move very fast if they want to. They're generally not dangerous to humans. They're much more finely built, much narrower in the body, and they're quite shy. So I once went swimming in a pool where I knew there were 40 freshwater crocodiles because we'd already caught them and released them. And the crocodiles stay out of your way. They're quite harmless. The saltwater crocodile, of course, is a completely different matter. Don't swim with those. But in these freshwater crocodiles, we found these tiny worms. And it took about 5 years to work out what the anatomy is. We really had no idea. What are the bits of this worm? What are we looking at here? So finally, a colleague I'd sent them to, who's an expert on the spirochetes, said he was looking at them one day and suddenly he realized what the anatomy, he suddenly understood the anatomy, and he instantly rushed out and grabbed the first person walking past in the corridor and made them come in and look. I think they must have thought he was a madman. But what do we have? Here is the male worm. There is the front of the male worm with the ventral sucker. Here is a large cavity inside the male worm. There is the front of the female worm. There is the oral sucker of the female worm. There is the ovary of the female worm. These are the yolk glands of the female worm on the side. The male worm has a cavity. The female worm sits inverted permanently, completely inside, completely enclosed by the male worm. We were able eventually, oh, there's a testis of the male worm. We were able eventually to find a tiny opening about here. So presumably through that, the female worm passes eggs out into the tissues of the crocodile. This is a very, very bizarre kind of parasite. So we have some DNA sequences from it now, and we really can't place it in any family. So this particular parasite still sits outside the classification of the blood flukes because we don't have a home for it. So there will be more work to do on that. So I said here, is it really a schistosome? Well, actually, we have no idea where it belongs. So this will be work for people to do in the future. And of course, schistosomes. Well, Apocalids kill fish, spirochetes kill turtles, schistosomes kill people. You all know that. You all know the causes and the disease that they cause. So I'm not going to talk about that in much detail. Only the family Schistosomatidae, the schistosomes, have separate sexes, male and female. And the male has a deep groove along the body. This is Schistosoma Manson Eye, I think, in which the female lies. So there is the front end of the female worm. Her body is lying inside this groove. Here is the rear end of the female worm. This anatomy, of course, is a reason for the family name. Schisto means split, and soma means body. Well, everybody knows about these. What people may not be so familiar with is the fact that maybe most species infect birds. And most species of schistosomes look rather more like these ones. They're very elongated. These are Trichobilharzia. They are the thickness of a hair and maybe 2 or 3 centimeters in length. They're extremely difficult to dissect from the tissues of a host. The other thing about most schistosomes is that that groove in the male is quite short. It doesn't occupy most of the body as it does in schistosoma. That's it. That's it there. Here in another close-up of the front of the male, there is that groove region, the gynecophoric canal or groove. So these worms are rather more typical of schistosomes. Many of the bird schistosomes, and particularly Trichobilharzia, occur in the nasal passages of water birds, in these turbinal scrolls, and they're in the blood vessels there. Rather similar to Schistosoma nasali, which lives in a similar place in cattle in India. And we assume that because ducks in particular often flush water through there, that any eggs which are passed out through the epithelium of the nasal mucosa can be washed into the water, or possibly they're swallowed and come out in the feces. We don't really know. But these schistosomes are extremely common in water birds. Do they occur around here? Of course they do. But we don't have much information on them. May be quite interesting to find out for various reasons, which I'll go into. And that is this. These bird schistosomes are extremely common, and some species, maybe all species, will penetrate human skin. A delayed hypersensitivity response is extremely common in humans. I certainly suffer from it, and many other people do as well. So this is an example of the kind of response that you see in people. I've occasionally infected myself to demonstrate to people just by putting some Secari and a drop of water in my skin, and you can feel them penetrating. And the next day, there will be a little papule there, and it will itch for a week. So is this important? Well, why should it be? Well, it is actually important for a number of reasons. One is that it makes it uncomfortable for some people to go swimming. In the Great Lakes of North America, Lake Michigan, the US state of Michigan actually set up something called the Swimmer's Itch Commission, a government department or government agency in the 1930s, because people in the middle of summer, it's very hot there, they wanted to go swimming in the fresh water of the Great Lakes. So this agency was charged with the task of checking for bird schistosomes. And they would say, this Acaria is starting to emerge. They would provide a forecast to tell people, don't go to these places on the lake because you will get this swimmer's itch. So people would either go further out into the open water or just stay at home. Nowadays, they go to Florida. So I think it's not such an issue in the Great Lakes, but the bird schistosomes are still there. The other thing, which is not commonly thought about, and perhaps it makes bird schistosomes the most neglected of the tropical diseases, and that is that there is serious potential for pathological effects. In some birds and in mice, these schistosomes will penetrate deeper into the body and actually penetrate the nervous system. Here is the schistosomulum of one of these bird schistosomes in the spinal cord of a mouse, 3 days after the mouse was exposed to infection. There are 1 or 2 people who are beginning to say, actually, this is potentially a serious issue in places where people are exposed to bird schistosomes regularly. There is one man in New Zealand in particular who is championing the cause of further investigation into this, because he says there may be cases of human disease which, well, we didn't know what caused them. It could be this, these schistosomes getting into the nervous system. So this needs further investigation. That I think is my arm here after exposing myself to some Secari in this lake in the north of Australia. Okay, I think you'll be able to leave by 10 o'clock. We have plenty of time. Schistosoma Manson Eye, of course, is the common blood fluke in Africa and South America and is mostly present as adults in the blood vessels around the gut. You may not know the history of this. It's present in South America because it was taken there during the slave trade over about 500 years ago, 500, 400 years ago. Infected slaves from Africa ended up in the West Indies, the Caribbean, and in parts of South America, carrying schistosoma Manson Eye. And it turned out there was a snail present in South America in which they could develop. Well, how did that come about? Because these things are supposed to be very snail specific. How come South America has been separate from Africa for, what, 70 million years or something like that? So somebody did some investigating and they discovered something rather extraordinary, that the snails in South America had come from Africa recently, but they hadn't come with the slave trade, they had come earlier. And they did a lot of work on this, and they found that on maybe several occasions, somehow freshwater snails from Africa had been washed into the sea from Africa in big floods. And there had been a little sort of pool of fresh water with weeds and things that somehow had been able to drift all the way across the Atlantic into South America with the snails still alive. And this probably happened, they thought, about 5 million years ago. So somehow the snails were able to move across the Atlantic and re-establish themselves in fresh water in South America. So that's an interesting point about the way in which this fluke was able to colonize South America. Then, of course, we have Schistosoma japonicum, which is zoonotic, and humans are not the only host, which means it's very difficult to eradicate. So Schistosoma japonicum is present in farm animals. This is photographed in Sichuan province, where Schistosoma japonicum is still present in people, and in water buffalo, and in rats, and in dogs, and it can infect rabbits and a few other species of mammals. So that's a problem for control. There has been, in this part of Sichuan, a massive control campaign. And I'll say a little more about that in a little while. Because around 2007, there was an effort made to reinvestigate the areas where the control campaign had taken place and to see whether the schistosomes had been able to re-establish. So schistosomiasis largely remains a disease of poverty. This photograph was taken in Lake Chad in West Africa. This lake almost no more exists. It's a very large lake, extremely shallow. The lake moves. It's so shallow that the lake moves if the wind blows in the other direction. The ground around it is extremely shallow. The entire lake can be pushed by the wind in various directions and is full of islands of reeds. And on those islands, there's a local group of indigenous people live, and they make boats with the reeds, and they make houses with the reeds, and they live by fishing, and they have Schistosoma hematobium. But the lake has almost vanished because too much water has been taken out by humans. This photograph is another poor area outside Lagos in Nigeria. Fresh water, lots of snails, lots of people, lots of schistosomes. And that's me at the age of 19, catching snails and fish and Schistosoma hematobium in West Africa. What's that? I didn't wear boots, no. Straight in the water. So I got a good dose of Schistosoma hematobium. And then I had to get it treated, so okay. Well, I told you I caught schistosomes. I meant that seriously. One of the things that has aided transmission of schistosomes in some places has been the construction of super dams. Well, in the whole of Africa, there was Kariba in what's now Zimbabwe and Zambia, that area. And this was the Akosombo Dam on the Volta River in Ghana. So it had been formerly a flowing river, where the main public health problem was onchocerciasis, caused by Onchocerca, a phalaroid nematode, which was transmitted by biting black flies. Well, the black flies bred in the flowing river. When the dam was built, the level built up, the water stopped flowing, no black flies, no Onchocerca. However, snails then started growing around the edges. Now we have schistosomes. Unintended consequences. I have to say, I've worked in some of the small rivers running into this dam where the black flies still breed. And the black flies, when they hatch and rise, it is terrible. I was once caught in a rise of black flies when they newly emerged. My whole body swelled up with hundreds of bites. It's terrible. And the disease, if you get the Onchocerca infection, is terrible. There were large regions of Ghana where the Volta River was running through, where everybody in every village over the age of about 15 was blind because of onchocerciasis. And this was regarded as normal by the people living there. Their children could see. Their children were going to go blind, but they were not yet blind. And there are photographs of children taking their parents out to the fields to work in the fields. So the parents would be able to feel to do the work, and the children would be able to guide them. It's just a terrible condition. So during colonial times, those villages, people were simply moved out of those villages. Those areas were just abandoned because of the black flies and onchocerciasis. And the photograph on the right is the region of the Yangtze River, which is now dammed by the Three Gorges Dam in China. There was little schistosomiasis in that area previously. The dam is now a good place for snails to live. At present, schistosomiasis seems not to be a problem there, but there is potential for it to be reintroduced. And I've made this point in a number of talks, so I'll say it only briefly. Schistosoma and humans did not evolve together. Schistosoma had already split into most of these species before humans even evolved. And then, on several different occasions, were able to jump from their previous hosts into humans. Schistosoma japonicum in East Asia, and possibly Malayensis and Mekong Eye were already there before humans came along, but we're not sure. But now these infect humans. Malayensis is almost gone. Schistosoma Manson Eye, rodents and similar things in Africa, jumped into humans. And the only group that appears to have evolved with humans might be some of the Schistosoma hematobium group. And members of this group in Africa are all able to hybridize with each other. And it's a very complicated arrangement, very complicated situation of parasites there. There are some more Schistosoma species which I'm not included on there, but these are the main ones which we need to worry about. So Schistosoma species jumped in different places at different times into humans and became human parasites. Also, at some point in the past, they switched their snail hosts. There was a big switch from pulmonate snails to a completely different group of freshwater snails, the cenogastropods. They jumped between those at some point in the past, millions of years ago. So they were able to falsify the hypothesis that they can never switch snail host. This was another long-range jump undertaken by these parasites. We don't need to go into the evolution of them much, except that hippopotamus might have been involved. And there's a happy hippo with schistosomes. This region between Africa and India in the Middle East during the Miocene 20 million years ago was mostly low-lying wetland. And there were a number of hippo species. And this possibly was a region where schistosomes could move between Asia and Africa. but we need a time machine to be able to go back and tell us. But what about Thailand? These species are known from Thailand. Two strange species from mountainous areas in the north of Thailand, which go through snails of the genus Tricula. These are the smallest of all freshwater snails. You almost can't see them. They're thin, 2 or 3 millimeters long when they're full grown. So these are the hosts for these. You'll find them in mountain streams around Chiang Mai. Spindali incognitum indicum herinasute, all in cattle and similar things. Spindali is known to cause human dermatitis, and indicum is known to cause human dermatitis, similar to the swimmer's itch. There was recently a paper, I'm not sure how to pronounce it, Krylas et al. 2022, describing cases in cattle and humans, that is, dermatitis cases in humans. There's also nearby, sort of in Thailand, not quite, Malayensis and Mekong Eye. And the only bird schistosome described from Thailand is Trichobilharzia magrethae. This causes dermatitis in humans. I am convinced that there are many other species, in birds in particular, in Thailand, which are able to cause human dermatitis. So there's a project for a master's student, perhaps. And will any of these species in Thailand eventually adopt humans as definitive hosts? Well, schistosoma species have done this in the past. As you saw already, they will do it in the future. And my candidates, my main candidate, will be Schistosoma indicum. Probably this has caused human schistosomiasis with mature worms in India. There's one village where this was present some years ago. It was probably Schistosoma indicum, but it was too long ago before we had DNA methods. Nobody is sure what caused it. People said it's hematobium, but it wasn't hematobium. That was in India. My money is on this species and possibly Spindali to jump at some time in the future between cattle and humans. Why? Because humans are in the rice fields, cattle are in the rice fields every day. These will jump at some point in the future. Will it be tomorrow or 1,000 years from now? I can't say. Well, I've got 10 minutes for an update. Let's start with genomes. That's mostly where the interest has been in recent years. And the first schistosoma genomes were published in 2009 in Nature. They even got a cover photograph. Look, front cover of Nature. How good is that? That was Schistosoma japonicum and Schistosoma Manson Eye, published by a group in China and by the Sanger group at the Sanger Wellcome Institute. At the same time, back-to-back papers in the same issue. So that was good. Of course, it was possible to identify a lot of genes in those genomes, but what do those genes do? Well, we're not really sure. We can think they have these kinds of functions. That was the early days. Today, GenBank has 25 full genomes of schistosoma species. Most of them were added last year. So this is recent and ongoing, and most of them from the Wellcome Sanger Centre in the UK. There's a lot of work going on there, and it's increasing all the time. So now we can start asking much more detailed questions. So from the original, oh yeah, we think these genes are involved in biological regulation. What's that? And these ones are involved in metabolism. Yeah, can't you be more specific? Well, back then, no, you couldn't. Now you can. These are only some chromosome level maps for a range of schistosoma species published last year. So schistosoma species have 7 somatic chromosomes, 7 pairs, and they have 2 sex chromosomes. These are the Z and Y chromosomes. Unlike mammals, the females have a Z and a Y chromosome, and the males have 2 Z chromosomes. So now people have mapped individual genes to the chromosomes. And they're available for a whole range of schistosoma species now, not just these 2. Well, what can we do with those? And I'm going to go quickly. Well, we can ask all sorts of questions. We can establish transgenic schistosomes and try to probe the function of particular genes. What happens if we put a gene in? What happens if we silence a gene? We can start targeting individual genes. And you may recognize some names there amongst the author list. There's a few familiar names. This one was published this month. Another case where they used knockdown methods to silence a gene, which was producing a substance necessary for cercaria to penetrate the skin, and which appeared to act in some kind of immunomodulatory manner so that the schistosome penetrating the skin could avoid the immune system. Another one, you may recognize some more names there. This was published in 2019, and they used a CRISPR-Cas9 approach on the eggs of Schistosoma mansoni to try to get transgenic eggs. This paper was published in the journal eLife, which I mentioned in a talk the other day, and it's already been cited about 78 times since 2019, which is pretty good. Cell Reports, published last year in Cell Reports, and they looked at Schistosoma japonicum. And they looked at Schistosoma japonicum from the 6 localities where it used to exist in East Asia, and found that these were extraordinarily different in their genetic makeup, as you can see on this principal components plot here. Those populations, the Sichuan population still exists, but has been knocked down to small levels. The Anhui population is still quite serious. The Japanese population no longer exists in the wild, only as a research population. Taiwan, maybe there's some, but most are only in a lab. Philippines is still a major problem. Indonesia, the island of Sulawesi, probably also gone, eradicated. So these 6 populations, there's probably only about 3 now that actually still exist. But they found that these are all very different. And this particular study, which I think is almost the last slide, published in 2021, was a follow-up from the kind of work that I tried to start in 2007. They went to the place in Sichuan where eradication had been attempted, went back and found that the worms were coming back. They were infected people. So the questions were, are these imported worms coming from Anhui, from the other place where we know it occurs? Or were these worms which had escaped eradication, which had survived through the mass drug administrations and the eradication programs? That was the question that I asked in 2007, but we were unable to answer it at the time. We tried. These people, and mainly Elizabeth Carlton, picked this up and went with it, and they were able to demonstrate that some of the worms had survived the eradication program. The small number of worms had come back, and we're now starting a new outbreak. And these new infections were due to a few surviving worms that had escaped eradication previously. Evidence, and it's not the first time that this kind of thing has happened in parasite eradication programs, that you can't say, oh, it's down to 1% now, so we can stop spending money on it. The problem's solved. Not at all. The problem is not solved until you have eradicated all of them forever. And you probably need to keep on checking and eradicating for many years. And an eventual goal of that should be complete eradication, but we're a long way from that. Also published last year in eLife. And of course, nowadays we're also interested in microbiomes and the holobiont, the whole thing. The helminth, the host, the microbiome of the host, the microbiome of the helminth. Now, work on exosomes lags behind similar work that's being done on nematodes, but work on Opus dorcas, which many of you are interested in, lags even further behind. So there are models for people to focus on for future research in metagenomics perhaps, and other things, by using models from work on schistosomes and on nematodes, on this, the holobiont, the whole thing, the whole organism, and all the things that live in it. So this was published also, well, I managed to get a copy yesterday. Anyone that's interested, I can send them a copy of this. Also brand new, just published. And the last slide is a picture of me in 2007 in Sichuan, demonstrating to people there that yes, you can pick individual miracidia of Schistosoma japonicum and use them for genetic studies. So I was demonstrating that you can take a single miracidium from the water and you can put it on some FTA paper and later on extract DNA from it. That's the last slide. Thank you, everybody.